Hello friends, till now we have seen the theory of parasitology, we have seen intestinal nematodes and the tissue nematodes in YouTube video lectures, but that was the PowerPoint presentation. Today we are taking the practical, which is of demonstration of various Helminths. So today we are starting that practical. In this crisis we have to devise the new techniques by which we can cope up with our studies, because it is said that when going gets tough, the tough gets going. So let's start today's session. So first we will see the adult worms. You will be given some specimen, you are supposed to identify them. So first we start with cystode, because in Helminths we mainly deal with cystode, trematodes and nematodes. So cystode I am putting here, giving the focus so you can see the morphology of cystode. You can see that it is a flat and tapped like worm. It is a flat like a ribbon, and it has many proglottids. This one is smaller, while if you see the tinea, then it would be a very long worm, and its proglottids would be expelled in the stool. It would be excreted in the stool. So that is the flat worm. Now we are taking the second one, and that is hydrated cyst. See, hydrated cyst is basically dog tapeworm. The adult worm would be in the dog, and human being is an accidental intermediate host. So in human it would produce such cyst. You can see the ball-like thing. So that capsular cyst would be formed into various organs. So liver being the most common organ, but it may occur in other organs like lung, spleen, kidney, even bone also. So that is cystode. Now the second, which is known as trematodes, or which are also known as flukes. So if I show you the fluke, you can see the size, and it is a leaf-like. Its name is trematodes, means it has a hole inside the body. With it would attach to its host. So this is liver fluke. So it is an example of trematode. And the third and the most developed worm is the nematodes, which are cylindrical in nature. So I am just showing you. You can see this is Ascaris Lumbricoides. So it is one of the largest intestinal nematodes. It may measure up to even 20 to 30 centimeter, and female worm is usually larger than the male one. So this is Ascaris Lumbricoides. Other than this, there would be other nematodes like hookworm and the threadworm. If you want to see it, I'll have to zoom it because they are very smaller, around 1 centimeter of the size. So I'm just showing you.
Cool.
Okay, now you can see this. This is hookworm. The size is around 1 centimeter, and it would remain attached to the intestine, and that would suck the blood, one of the common cause of anemia.
Cool.
This is threadworm. They are piece of thread like. Now it is settled. I am moving it, so now you can see that it is looking like the pieces of the thread. Clear? So they are much small, even smaller than the hookworm, less than 1 centimeter of the size.
Cool.
So after adult worm, how to identify the eggs of the Helminths? When you do the stool preparation, you will see the eggs of the helminth. So I am just showing you the common helminthic eggs. Now you will notice here that some of the eggs would be yellowish brown in the color, and some are colorless. So the eggs which take the bile from the intestine would give this color, and so these eggs are known as bile stained egg, while those eggs which look colorless are the non-bile stained egg. And this is the relative size of the helminthic egg. For example, this is the size of RBC. So eggs of cystosome and other nematodes are much bigger, while intestinal nematodes are slightly smaller than that. Now, if we see one by one, the common eggs which we encounter. See, this is tinea. The egg of tinea. Here, by looking at the morphology of egg, you cannot discriminate between tinea solium or saginata. Both would live same in that it would be bile stained. There would be radial striation which would be visible in the high power of the microscope. If we see next egg, this is the round one, the egg of Ascaris lumbricoides, which is commonly seen in the stool preparation. There is a rough boundary. It is yellowish brown in the color, and you can see in a fertilized, you will see ovum inside. The next egg, if we see, which is again bile stained, this is known as egg of Trichuris ticula. The unique feature of this egg is you can see that there would be at both the end there would be polar mucus plugs. So that is a characteristic barrel shaped egg with these two mucus plug, that is Trichuris ticula. Next, this is hookworm, another very common intestinal helminth, one of the major cause of anemia in developing countries. Now you see this egg, the size is around roundworm only, around 80 to 60 micrometer, but it is non-bile stained, and you can see a clear shell and desegmented ovum. And the last egg which we see is this. This is egg of threadworm or the pinworm, that is Enterobius vermicularis. The peculiar characteristic of this egg is its shape. It is known as plano-convex shape. One side is relatively flat and one side is convex. And this egg can be seen in the NIH soap, because this egg are laid on the perianal skin, and in NIH soap it would stick to the cellophane tape and you can see that. So with this we finish today's demonstration of various parasitic worms as well as its microscopic structure. Hope you have enjoyed this presentation. You can in a last note to this, in this corona season, I remember the lines from the Gujarati poet Sri Shekhadam Abuwala. એમણે એમની પ્રેમિકાના સંદર્ભમાં કીધું છે કે પૂછતી નહીં તારા વિના કેવા અમારા હાલ છે, પૂછતી નહીં તારા વિના કેવા અમારા હાલ છે, આંસુઓ છે આંખમાં અને હાથમાં રૂમાલ છે. આ જ વાત આપણે થોડી પંક્તિ સાથે છૂટછાટ લઈને કોરોનાના સંદર્ભમાં કહીએ તો પૂછમાં તારે લીધે કેવા અમારા હાલ છે, પૂછમાં તારે લીધે કેવા અમારા હાલ છે, સેનિટાઈઝર છે હાથમાં અને મોઢા ઉપર માસ્ક છે. So keep studying. If you have any comment or queries, please feel free to ask us anytime. Thank you very much.